Bismillahirrahmanirrahim. Welcome my friends. Welcome to another video lecture by Kami Biologies. In a series of video lectures, we are going to talk about antifungal drugs in a detail. Now, this is the first video lecture regarding with the antifungal drugs, and in this video lecture, we are going to talk about a basic overview, or we can say we talked about the big picture of the antifungal drugs. Now, before going to explain the antifungal drugs, first you need to understand the fungi cell membrane or fungi cell structure. We can say once you get idea about the fungi cell structure, so then everything, or we can say the drugs the drugs mechanism of action of the drugs will be very easily for you. Fine. So now in this video lecture, what we are going to talk about, we are going to talk about basic overview, or we can say the big picture, basic overview of antifungal drugs. Antifungal drugs. Now, antifungal drugs are those agents which can kill the fungi, or those agents which can stop the growth of fungi. So let me write. They can either kill or they can stop the growth. Now, before going to explain the drugs which are used in antifungals, first of all, we should have to understand about the structure of the fungi. So once we get idea about the structure of the fungi, so once we understand about the fungi structure, so then after that I will write the drugs which are used in antifungal drugs. Fungi are unicellular as well as fungi are multicellular. Multicellular fungi we call that filamentous, and unicellular fungi we call that yeast. So let's talk about the unicellular fungi in this case. So what actually happen? Fungi will be some fungi will be in a circular form. Some fungi are present in elongated rod form. There is a lot of structure, or there is a lot of shapes of the fungi. So this is not a correct one shape, but it will just clear your concept. Now this outer layer is, let's suppose we call that what? We call that this is the cell wall of the fungi. Now this is the cell wall. This inner layer, let me draw with the red marker, this is the cell membrane. Fungi have cell wall as well as cell membrane. At the mid portion there is a nucleus, and inside the nucleus there will be a genetic material. Now, as all of you better know that fungi are eukaryotic cell. So all organelles which will be present in the fungi as in our own cell as well, because in our own cell, if we see, it is also eukaryotic cell. So in our own cell they will contain mitochondria, ribosomes, Golgi apparatus, endoplasmic reticulum. Everything will be present in the fungi as in our own cell, but the composition of each of organelles will be different. So there will be ribosomes, endoplasmic reticulum, and everything as present in the eukaryotes. I am not going to talk about that, because here I will just talk about the antifungal drugs. Now, so this is the nucleus portion. Inside the nucleus they will contain the DNA, and then the rest of the organelles will also present. First of all, so that's all about the structure. First of all, let me draw the cell wall in a detail. Not in a detail, a little bit. Then I will draw the cell membrane, and then I will draw all the organelles one by one, a little bit. Now what actually happened? First of all, let me draw the cell wall. A fungi contain a cell wall which can give rigidity and the flexibility to the fungi cell. Now this cell wall actually contain what? We can say beta 1,3 and beta 1,6 glucan. This blue one represents the beta 1,3, let's suppose, and this green one represents beta 1,6. So they contain, just like the cell wall will be just like in this form. Now this is the cell wall of the fungi. We can say that how this means, we can say that how all these will be synthesized. Now there will be an enzyme. Let me draw here the enzyme structure. We usually called it beta glucan synthetase enzyme. So enzyme will be look like in this form. Now this is beta glucan synthetase enzyme. This enzyme, there will be small molecules. Let me draw here. And let me also draw with the green marker as well. These small molecules we call it beta 1,3 and beta 1,6 glucan, immature beta 1,3 and beta 1,6 glucan. These immature beta 1,6 and 1,3 glucan will passes from the enzymes we call it beta glucan synthetase enzyme. Once they pass from this enzyme, then they will convert these beta 1,3 and beta 1,6 into a mature beta 1,3 and beta 1,6 in a chain form. So then we will get this chain form like structure, and this is actually a higher, and this is actually the chitin, we can say. So we can say that the fungi cell is made, the fungi cell wall is made up of chitin. So all these material are actually what? All these material are actually made up of the chitin. How? Inshallah, I will discuss this cell wall more in detail in our next video's lecture. Now, another important thing which are present inside the, after the cell wall, there will be another things. So let me draw with a black marker. Now this is called what? This is called cell membrane. Inside the cell membrane there will be a steroid present. We usually call this steroid what? We usually call this steroid agasterol. In case of eukaryotic cell, or in case of our human eukaryotic cell, it contains the cholesterol, but in fungi this contain will agasterol. So cholesterol will be replaced into agasterol in case of the fungi cell membrane. So this is the cell membrane. Due to this agasterol, it will provide flexibility and rigidity to the bacteria, to the fungi cell. Fine. So this is another thing. Now we can say that how this agasterol will be synthesized. This agasterol will be synthesized. We have an enzyme, we simply call that. I'm not drawing the detailed structure of the enzyme which are used in the synthesis of the agasterol, but I will just a little bit explain it. So this is, let's suppose, an enzyme. We called it 14 alpha demethylase enzyme. They contain cytochrome P450 also. They contain a cytochrome. Let me draw with a blue marker. Cytochrome P450. So due to this enzyme, there will be a Lensterol. There will be a Lensterol molecules. These are what? These are the Lensterol molecules. These are the Lensterol molecules. And there will be another kinds of molecules. Let me draw with the red markers. These are another molecules. Now, what actually happened? We call that squalene. These are the squalenes. First of all, what will actually happen? These squalenes will passes from an enzyme. Now, these squalenes will be passes from an enzyme. We usually call it squalene monooxygenase. So, this is the structure of squalene monooxygenase enzyme. Now let me talk a little bit about that how agasterol will be synthesized. The agasterol will be synthesized that first of all these squalene molecules will pass from this squalene monooxygenase enzyme, and then from this squalene monooxygenase enzymes, this squalene will be converted into Lensterol, and then the Lensterol will be passes from the alpha 14 demethylase enzyme, and after that what we get? We get the agasterol simply. And so that's how the agasterol is synthesized. Now, so that's about the agasterol synthesis, and we can say that's the cell membrane of the fungi. Now, there are some other macromolecules, or we can say there are a special kinds of molecules. We call that macromolecules. These macromolecules are actually responsible for the fungi growth. So let me draw the macromolecules with these blue markers. So let's suppose these are what? These are the macromolecules. These molecules are actually responsible for the fungi growth. Fine. So now How this will be synthesized, inshallah, I will discuss it in a detail in next video's lecture. Now, so these are the macromolecules which are present inside the fungi. Inside the fungi, as I told you, there will be a nucleus also present. So let's suppose this is a nucleus, one portion. So inside the nucleus, what we have a genetic material, simply we can say a DNA. So this is a DNA. Now, this is another main important things which are targeted by the antifungal drugs. Now we have another molecules which are also present inside the fungi. We simply call that what? We simply call that polymerized macrotubules. Polymerized macrotubules. So let me draw here. So these are the polymerized macrotubules. Now these polymerized macrotubules are responsible to make a spindle fibers. So once they make a spindle fiber, so then the fungi will be divided into two cells, we can say. So these are what? These are the microtubules, or we can say polymerized macrotubules, that are responsible for the spindle fiber formation. Now, so that's another important thing that in which the antifungals are affected. So that's all about the important material, or although there are some other organelles as well, because all of you better know that fungi is what? Fungi is a eukaryotic cell. So inside the eukaryotic cell there is a ribosomes, endoplasmic reticulum, Golgi bodies, and some other organelles also. But I am not going to talk about that. Why? Because the antifungal drugs will not affect it on that area. The antifungal drugs will just affect it on these important sites. So that's why I explain it. Now let's talk about the drugs one by one. We have a drugs, we simply call that Pauline. We simply call that Pauline. Inside the Pauline, we have a successful drugs, we usually call that what? Amphotericin B. Amphotericin B. These drugs, what are the mechanism of these drugs? I will explain it in a detail, inshallah, in the next video lecture. But the mechanism of action of this drug is usually they can target it, this agasterol. So they can target it what? They can target it the agasterol. They can bind with the agasterol and make a holes by which the cell will be rupture, or we can say the cell will be die. So these drugs are what? These drugs are the fungicidal in nature. Now there is another drugs, so this is number one. There is another drugs, we call it echinocandins. Now let me write the name. These drugs are actually mentioned in the glucose synthetase inhibitor. Glucane synthetase inhibitor. And we usually call that what? We usually call that echinocandins. So we have echinocandins which are usually used in the glucane synthetase inhibitor. What are the mechanism of action of these drugs? Now the mechanism of action of these drugs can easily bind with this glucane synthetase enzyme. So once they bind with this glucane synthetase enzyme, so what actually they do? They will block. What they can block? They block the passage, or they can block the passage of this immature beta 1,3 and beta 1,6 glucane. So once these once the passages are blocked, then these beta 1,3 and beta 1,6 glucane will be not passed. So when they are not passed, so then we can say cell wall will be not synthesized. We have some other drugs, we usually call it fluorinated pyrimidine. Fluorinated pyrimidine. Fluorinated pyrimidine. The drugs which are used in fluoride, the successful drugs are the fluocytosine. Fluocytosine. Now this is the successful drugs. This fluorinated pyrimidine, we can say they can encrypted with this DNA. Fine. So they can encrypt with this DNA, and we can say that break down the DNA. So once the DNA is break down, so there will be no transcription and no translation process will be occur. Although these DNA are also present in a eukaryotic cell, but how these fluorinated pyrimidine, or we can say flow cytosine, will affect the DNA of the the DNA of the fungi, but not our own DNA. Inshallah, I will discuss it in a detail in some other videos lecture. Now, so these are the third number drugs that we are going to talk about. Now, the fourth drugs which are used in this case, we usually call that, inside azole we have what? We have imidazole. Imidazole. Now, what are the mechanism of action of these drugs? The mechanism of action of these drugs, they can block or they can strongly bind with that area of this 14 alpha demethylase. So they can bind with the 14 alpha demethylase enzymes. So once they bind with the 14 alpha demethylase enzyme, this lensterol will be not converted into agasterol. So this is another drug. This is the fourth one. The fifth drugs that we are going to talk about, we are called that what? We call that, let me erase this one, pyridone. Pyridone. Now the drugs which are used in pyridone, we usually call that cyclopyroxolamine. Cyclopyroxolamine. This spelling may be right or wrong. So this is actually not the correct spelling. Fine. So this is what are the mechanism of action of these drugs? These drugs can stop the synthesis of these macromolecules. Fine. So they can stop the synthesis of macromolecules. So once the macromolecules are not synthesized, so the fungi growth will be halted. So these drugs are what? These drugs are fungistatic in nature. This amphotericin B, or we can say choline, are fungicidal. The second one are also fungicidal, means echinocandins. This third one drugs are fungistatic in nature. This fourth one are also the we can say this fourth one are also what? These are also fungicidal in nature. This fifth one are also fungistatic in nature. Now the sixth one drugs that we are going to talk about in next videos, we usually call that allyl amine. So these are the other drugs. Inside the allyl amine, we have the special kinds of drugs. Let me write the one name: terbinafine. Now, what are the mechanism of action of this allyl amine? The mechanism of action of this allyl amine, if we talked about the mechanism of action of this allyl amine, so they can strongly bind with this one enzyme and stop the activity of this one enzyme. We call it squalene monooxygenase. So they can bind with the squalene monooxygenase enzyme. So once they block the squalene monooxygenase enzyme activity, so then the squalene will be not converted into lensterol, or we can say the pathway of the agasterol synthesis will be halted. Fine. So these are the allyl amine drugs. Now the last one drugs that we are going to talk about, these are the, let me write here because I don't have any other space. We call it the grysofluene. Grysofluene. Now what are the mechanism of action of this grysofluene? These grysofluenes will bind, or they can attack on these micropolymerized microtubules. So once they can bind to this polymerized microtubules, so then spindle fibers will be not formed, and after that the cell will be not divided. So it is also fungistatic in nature, and this allyl amine are also the fungicide in nature as well. These will be our, so this one drugs will be our last videos lecture. Fine. So I hope all of you will understand about that. So Inshallah, in the next video lecture, we are going to talk about this proline, or we can say amphotericin B, in a detail, the mechanism of action, resistance, side effect, and antifungal activity of the antifungal spectrum of these drugs in a detail. Fine. So if you like all these videos, so if you like this video lecture of the basic overview of the antifungal drugs, so then please hit the like button, and also share this video with your friend, and subscribe the channel to get more updates of these videos lecture. Fine. Thank you. Thank you so much.